Hello, this is Dr. Nanda Prasad, Professor, Oral Pathology, Sri Rajiv Gandhi College of Dental Sciences and Hospital, Bangalore. Today I will be talking about stem cell research and dental pulp stem cells. The purpose of this presentation is to make the audience conversant and comfortable with the basic process and terminologies used in stem cell research and dental pulp stem cells by the end of this lecture. So, what are these stem cells? These stem cells are special human cells that are able to develop into many different cell types. They can range from a brain cell to a muscle cell. In some cases, they can repair damaged tissues as well. Now, there are some terminologies used in stem cells. Now, what are these clonogenic cells? What do you mean by these clonogenic cells? These clonogenic cells refers to the fact that these cells are clones of one another. Now, what is unspecialized? The fact of the matter is that they can divide and give rise to different specialized cells. What is this capable of self-renewal? What do you mean by that? It is a process by which stem cells divide to make more stem cells, perpetuating the stem cell pool throughout life. What do you mean by multilineage differentiation? Multilineage differentiation is the ability by which to proliferate, it's a dual ability actually, to proliferate indefinitely and differentiate into multiple tissue types. So, to sum up, stem cells are essentially progenitor cells which are capable of regeneration and differentiation into a wide range of specialized cells. Now, what is the types of stem cells? We'll just get ourselves used to these terminologies. What is totipotent? Totipotent cells can form all the cell types in a body, plus the extraembryonic or placental cell. It is found that during cell divisions in embryonic development that more totipotent cells are produced. Totipotent cell has the capacity to form an entire organ. An example of that would be a fertilized ovum. What is pluripotent? They are master cells capable of making cells from all the three basic body layers like the mesoderm, ectoderm and the endoderm, so that they can potentially produce any cell or tissue the body needs to repair itself. This master property is called as pluripotency. Then what do you mean by multipotency? These are stem cells that are capable and have the capacity to self-renew by dividing and to develop into multiple specialized cell types present in a specific tissue or organ. Now, types of the other stem cells. What is embryonic stem cells? Embryonic stem cells are stem cells derived from the undifferentiated inner mass cells of a human embryo. They are pluripotent and totipotent too sometimes. Adult stem cells, also called as postnatal stem cells, they are undifferentiated cells found throughout the body after development, and they multiply by cell division into and replenish dying cells and regenerate damaged tissues. Also called as somatic stem cells, they can be found in juvenile as well as adult animals and humans, unlike the embryonic stem cells. Now, going on to a diagram which just shows you what pluripotent stem cell does. You can see the fertilized eggs forming the 8-cell embryo, going on to form the blastocyst, then displays pluripotency here and forms the neural cells, cardiac muscle and the blood cells. Now, going on to a little bit of historical background about this stem cells. As early as 18th century, people started talking about benign embryonic beginning, and for the next 100 years there was silence. By 19th century, they started talking about discovery of the bone marrow stem cells. The people didn't just sit quiet over this. They started their experiments. Some people started doing a bone marrow was given by oral route to treat anemia and leukemia. Obviously it ended in failure. They didn't know why it failed also. Then they tried animal experiment with bone marrow transplant. Again the same result. Allogenic bone marrow transplant in humans were tried. Failure. They didn't know what the cause was all about. But as late as 1958, a French person called Jean Dosset discovered human histocompatibility antigen, or the HLA factor. This would turn things around for stem cells. In 1960, significance of HLA in the success of transplants was acknowledged. Now, what is this HLA? HLA, these are cell surface proteins which are responsible for the regulation of the immune system. So, as they started developing new things, by 1990, rapid expansion and success of bone marrow transplants was seen. In 1998, a person called James Thompson developed the first embryonic stem cell line. John Gerhardt worked on this and developed germ cells from the fetal gonadal tissues. Now, where do you get this embryonic stem cells? Issue started, where do you get it? So, in vitro fertilization of the embryos from the inner cell mass of blastocyst, you would get the embryonic stem cell. Then again you would get it from the primordial germ cells from the fetal gonads too. So, what are the advantages of embryonic stem cells? If you're doing any procedure, there should be an advantage as well as a disadvantage. You need to know that. The advantages of an embryonic stem cell is, one is its flexibility. Its potential to make any cells was seen. Immortal, endless supply of cells with defined characteristics was seen. Availability, embryos from the IVF clinics. You could go into any IVF clinic and get an embryonic stem cell because they were available. There has to be disadvantages of embryonic stem cell. Destruction of human life. Definitely an ethical and moral issue was there. You couldn't destroy human life and extract embryonic stem cells. Then difficult to differentiate uniformly and homogeneously to a target tissue. Suppose they targeted a tissue and wanted a growth, they couldn't get the desired result as far as embryonic stem cells were seen. Now two concepts started troubling them. One was the immunogenicity and other was the tumorigenicity. What is this immunogenic? What do you mean by that? It is simply defined as the ability of a cell to induce an immune response that can prevent its growth. But our understanding of this mechanics is still evolving. Now, what is this tumorigenicity? That is a tendency to produce tumors. These were the disadvantages of the embryonic stem cells. The sources, where do you get this adult stem cells? One's from the pregnancy related tissue, example, the umbilical cord, the placenta and the amniotic fluid. From these they were harvested. From the adult tissues, you would get it from the bone marrow, liver, epidermis, retina, brain, skeletal muscle, intestine and the dental pulp as well as the periodontal ligament. So, sources where even you would be able to harvest adult stem cells from the cadavers too, provided that within 20 hours following death, if you are able to harvest, you would be successful. Example, the neural stem cells were harvested this way. So, advantages of the adult stem cells. The flexibility is pretty similar to the embryonic stem cells and inducing, easy to induce, that's what I meant. The relative ease of procurement. There, one more thing you should observe here. Unlike the embryonic stem cells, the problems they encountered there, here they wouldn't encounter as far as the non-immunogenic and non-tumorigenic. They are non-immunogenic as well as they are non-tumorigenic. They never found a problem involving the immunogenicity and the tumorigenicity here. So, what are the disadvantages of adult stem cells? Limited quantity, difficult to obtain in large numbers. It would be very difficult to obtain in large numbers. Finite, may not live as long as embryonic stem cells in culture. Difficult to reprogram into other tissues. These were the problems they encountered when they were dealing with adult stem cells. So, what is this uses of basic stem cell research? You wonder, why are you doing all this? What are the uses? One is the basic medical research. Stem cell research is the area of research that studies the properties of stem cells and their potential use in medicine. Or you could say to understand the genetic and molecular controls of cell division and differentiation in disease. So, and to test the safety of new drugs too. With toxicity screening, drug companies started keeping banks of stem cells from a wide variety of genetic background so that they could test on how organ cells created from those stem cells reacted to a drug, thus removing drug testing live human candidates. Cell-based therapies were done since 1970s. Stem cell therapy using tissue stem cells was seen. Parkinson's, spinal cord injury, burns, heart disease, diabetes, and osteo and rheumatoid arthritis were treated using this. So, let's go on to some other terminologies used in this chapter. What are these terminologies? You would encounter these terminologies. One is, what do you mean by in vitro or ex vivo? It means outside the living body and in an artificial environment. In vivo, studies conducted inside living organisms. Then what is an explant? It is a process by which a piece of tissue has been transferred from animals and plants to a nutrient medium. Now what is an implant? Implant is a stem cell engineered matrix which was found to promote cell proliferation within the body. Now what is a transplant? A donor stem cells are transfused into a patient's blood so that they go to the bone marrow. Prior to this, the patient receives a very high dose of chemotherapy and radiotherapy. Now, some other terminologies you need to know. What is a cell line? A cell culture developed from a single cell and therefore consisting of cells with a uniform genetic makeup, and their permanently established cell cultures. Or you could say a permanently established cell culture that will proliferate indefinitely when given appropriate fresh medium and space. Now what is passage? You need to know there are two kinds. There is an early passage stem cell and there is a late passage stem cells. In early passage stem cells have high differentiation ability compared to the late passage stem cells, or you could say it's a process of passing or maintaining a group of cells through a series of hosts or cultures. Now going on to the potentials of dental pulp. As you all know, pulp is a loose connective tissue of ectomesenchymal origin. It provides dentinogenic, sensory, nutritive and defensive functions to the tooth. It also has a reparative dentinogenic response to disease and injury by a progenitor or a stem cell. So going on to the dental pulp stem cells. What are they? They are mesenchymal stem cells that have the ability for multipotent differentiation as well as self-renewal ability. They have been useful not only for dental disease but also for systemic diseases too. So what are these things about dental pulp stem cells? There is something called dental stem cells from human exfoliated deciduous teeth, also called SHED. What are they? They are highly proliferative pluripotent cells that can be retrieved from primary teeth. They are isolated from physiologically shedding deciduous teeth, and the way of harvesting is minimally invasive technique of retrieval is seen here. So what happens here? So there has to be a method of extracting it. Before I go on to the method, one thing I'll have to tell you is these SHED cells or the stem cells from human exfoliated deciduous teeth, they can be cryopreserved and stored for long periods of time. So you might ask me, how do you extract it or how do you harvest it? There are two techniques at this level. I would just mention what these techniques are. One is the enzymatic digestion technique, other is the tissue explant technique. What is this enzymatic digestion technique? Digested minced pulp tissue is mixed with type 1 collagenase and dispase and again mixed with the enzyme. Then what is this tissue explant? Minced tissue pulp are placed in tissue culture dishes, allowing the outgrowth from the culture. This is about the tissue explant. Now, what is this multipotent property? What we are talking about being multipotent? They are multipotent mesenchymal cells. They possess distinct properties when compared to other mesenchymal stem cells. So, we are talking about higher proliferation rate. What is this higher proliferation rate? They have been shown to have higher proliferative rate when compared to the bone marrow. So when these SHED cells, stem cells were studied, they found that they exhibit high ability to proliferate and differentiate. Studies have shown that they are capable of generating robust amounts of bone and pulp and the pulp dentin complex in vivo. In vivo, when they checked, they found that they produced robust amounts of bone and pulp dentin complexes. They had high osteoinductive capacity in vivo. Stem cells from the human adult teeth. The first stem cells isolated were from the adult human dental pulp. They were isolated from permanent third molars and exhibited high proliferation and high frequency of colony formation. They produced sporadic but densely calcified nodules. They had high rates of cell renewal. Now, talking about multi-lineage differentiation of this kind of dental pulp stem cells, they also have the potential to give rise to other cell lineage similar to but different in potency from that of bone marrow derived stem cells. They displayed capacity to develop three distinct cell lineages: the osteodontogenic, adipogenic, and the neurogenic. Now, formation of ectopic dentin and pulp in vivo was also seen. The pulp dentin complex formation was seen in vivo when they made an in vivo study. So, applications of dental pulp stem cells. This basic dental research, the basic dental research works on the premise that they have shown a potential or possibility for various human diseases, including dental related problems, or you could say to characterize the functional role of differentially expressed genes. The clinical dental research, what about that? To repair damaged tooth structure. For example, the dental pulpal stem cells are a promising source of cells for numerous and varied regenerative medicine applications. Their natural function is in the production of odontoblasts to create reparative dentin and also to support and induce bone regeneration applications in dentistry and also in the regeneration of the tooth structures. The orofacial tissues have been identified as a source and therapeutic target for stem cells with clinical interest in dentistry. And one more thing we have to say, the biggest clinical dental research application would be in dental stem cell banking. It is found to be a secure way to store your child's baby teeth, wisdom teeth, or healthy extracted teeth. So, what they say finally is bless your child with the best possibility for a healthy future. So, how would I like to conclude this? I would like to conclude by saying that we have observed that dental pulp stem cells displayed multilineage potential to differentiate into odontoblasts, osteoblasts, adipocytes, and neural tissues. In vivo cultures followed by in vivo transplantations have realized that they have been ideal for tissue reengineering in endodontics. It is clear that culture medium could be used to modulate the cell proliferation and differentiation. Thus, finally, we realized that there is a promise and a high clinical potential to provide an alternative to traditional pulp therapy followed today. Thank you.